Today we will talk about the supplementary genes. What are supplementary genes? Supplementary genes are two independent genes which interact to produce a new trait together, but each dominant gene independently can produce its own trait. This is what is called as supplementary genes. So we can say that supplementary genes are two independent genes. These are two independent genes which interact. These are two independent genes which interact to produce new trait. Together, but separately, alone, independently, each dominant gene, but alone, each dominant gene produce its own trait. So this is the definition of supplementary genes. That means supplementary genes are two different genes, and these two different genes, they separately, independently can produce their own trait, their own phenotypic trait, but when they are brought together, they interact and produce a new trait. So these are called as supplementary genes. These supplementary genes are non-allelic genes. That means they are two different genes. They are non-allelic genes. They occupy different gene loci on the chromosomes. These supplementary genes occupy different gene loci on chromosomes. So we have the examples of supplementary genes. So we'll take the example of interaction of supplementary genes in case of combus in fowls. Fowls are simply chickens. That means in case of combus in fowls, the supplementary genes interact to produce the combus in fowls. We will discuss these. In fowls, in chickens, there are four types of combus. So we have rose comb, we have pea comb, walnut comb, and single comb. So in chickens there are four types of combs: rose comb, pea comb, walnut comb, and single comb. This rose comb, it is produced by the dominant gene R, and this pea comb, it is produced by the dominant gene P. When these two dominant genes R and P are brought together, they produce a new comb that is called as walnut comb. And this single comb, it is produced by the two recessive genes, small r and small p. So it would be like this. So rose comb, it is produced by dominant gene R, and this pea comb, it is produced by dominant gene P, and this walnut comb, it is produced by the interaction of R and P genes. And this single comb, it is produced by two recessive genes when they are present in the homozygous state, that is small r and small p. So it would be like this. Rose comb, it would be capital R, small p, capital R, small p. So this is the genotype. And this P comb, its genotype P comb would be capital P, small r, capital P, small r. So walnut comb, it is produced by the interaction of two dominant genes R and P. So it would be like this: capital R, capital P, small p, and capital R, capital P. This single comb, it is produced by two recessive genes when they are present homozygous state: small r, small p. So it means that these two dominant genes R and P, they have interacted. R and P can independently produce their own trait. That means R produces phenotypic trait rose and P produces phenotypic trait P. But when these two dominant genes are brought together, they produce a new phenotypic trait, that is walnut comb in case of chickens. We will explain it with the help of a cross. We will take parents. Rose comb and P comb. Rose comb chickens, they will be crossed with P comb chickens. Obviously the genotype is capital R, small p, and genotype here is capital P, small r. This is genotype. Obviously we will get gametes, law of segregation, meiosis will operate. So we have gametes capital R, small p, and capital R, small p. Here same gametes capital P, small r. This is capital P, small r. These are gametes. Then we will get F1 generation. First filial generation is, we have capital R, small r, capital P, small p. So these are this individual, these chickens, these fowls, neither they are rose combed nor they are P combed, but these are walnut combed chickens. These are walnut combed fowls. It means that this R gene and P gene, they have interacted and produced a new trait together, new phenotypic trait, that is walnut combed trait. But independently, alone, they can produce their own trait. That means R can produce its own trait, rose comb, and P can produce its own trait, that is P comb. Then F1 individuals are crossed together, that means selfing. Here F1 individuals are crossed together. It would be like this: capital R, small r, capital P, small p. They are crossed with capital R, capital P, small p individuals. So we will make gametes. This is capital R, capital P, capital R, small p. These are gametes. Then we have small r, capital P, small r, small p. Same is the case gametes in this individual. We have capital R, capital P, capital R, small p, and we have small r, capital P, small r, small p. These are gametes. Then in order to get the F2 generation, there would be the fusion of these gametes. We'll explain it by means of a Punnett square. It's called a checkerboard. It will be like this. We have capital R, capital P, capital R, small p, small r, capital P, small r, small p. Same as these are gametes. This is male gamete. This is female gamete. These are gametes. So we'll get F2 generation by the fusion of these gametes. Here, female gametes, capital R, capital P, capital R, small p, small r, capital P, small r, small p. So these are F2 individuals. So we have this capital R, capital R, capital P, capital P. Obviously this is what? This is walnut. Capital R, capital R, capital P and small p. This is walnut because both dominant genes are present. Then we have capital R, small r, capital P, capital P. This is also walnut. Then we have capital R, small r, capital P, small p, walnut. Then capital R, capital R, capital P, small p. This is walnut. Capital R, small p, small p. This capital R, small p. This would be rose combed. This is rose combed. Then we have capital R, small r, capital P, small p. This is walnut combed. Walnut combed. Walnut combed. Then this is capital R, small p, small p. This is rose combed because the dominant, only one dominant gene is present, that is R. That is why it is rose combed. Then we have another capital R, small r, capital P, capital P. What is this? This is walnut combed. Then capital R, small r, capital P, small p. This is what? This is walnut combed. Then we have small r, capital P, capital P. That means only one gene, one dominant gene, capital P is present, so it would be P combed. And we have small r, capital P, small p. One dominant gene P is present, that means it is P combed. Then we have here capital R, capital P, small p. This is walnut combed. Then capital R, small p, small p. This is also rose combed because only one dominant gene is present, that is R, so it is rose combed. Then small capital P, small p. This is what? Only one dominant gene P is present, that is it is P combed. Then we have small r, and small p. That means two homozygous recessive genes are present, small r and small p. Then it is single combed. Then it is single combed. So it means that we have walnut, we have rose, we have walnut combat chickens, rose combat chickens, pea combat chickens, and single combat chickens. That means the ratio is walnut is 9:3:3:1. So 9 walnut, 1 2 3 4 5 6 7 8 9, 9 walnut. We have 3 rows, 1 2 and 3 rows. We have 3 pea, 1 pea combat, 1 pea combat, 2 pea combat, 3 pea combat. And we have what? 1 single combat. That means 1 single combat. So the ratio is 9:3:1. We can have walnut combat. It is out of 16, 9 are walnut combat. Then we have rose combat. Out of 16, 3 are rose combat. Out of 16, 3 are pea combat, and out of 16, 1 is single combat. So we can see this 9:3:1. This resembles with the simple dihybrid ratio. So we can say that the inheritance of rose genes and pea genes, pea combat genes and rose combat genes, they resemble with normal Mendelian inheritance. Only the difference between this cross and the simple dihybrid cross is that in the F1 generation, in this cross we get the F1 individuals. They do not resemble with either of the parents. This is the main difference between the simple dihybrid cross and this cross. In simple dihybrid cross, this F1 individual resembles with the parent, whereas in case of supplementary genes, the interaction of supplementary genes, the F1 individual does not resemble with either of the parents. That means here we have parents rose combat and pea combat, whereas in the F1 generation we get individuals which are walnut combat. This is because of the interaction of the supplementary genes. When these two supplementary dominant genes, they brought together, they produce a new phenotypic trait, but each dominant gene alone and independently they can produce its own trait. These are what are called as supplementary genes, and when they interact, these supplementary genes, when they interact, they produce a new phenotypic trait in the F1 generation. This was regarding the supplementary genes. Thanks.